Okay, let's get started. Welcome to day 2 of Spatial Transcriptomics workshop. I want to start by thank you for sticking around at this time. And also, I want to thank you for whoever came yesterday, including you guys, for your participation, questions, and discussion. It has been really fun yesterday. It helped make this workshop better in a way I didn't anticipate it. And I also learned something new. So that's going to be the first thing I'm going to share with you guys today. So can those online hear me well? Can you give me a thumb up or just speak up?
Yeah, we can hear you.
Okay, thank you. So let's get started. Yesterday by the way, if you need a grade, here's the attendance sheet, and it's also in your email. You don't have to copy this. It's in your email. So yesterday we did a lot of heavy duty thing, heavy lifting thing. Why we need a spatial transcriptomics, what it is, how does it work? We read some research paper, and we did some coding exercise, although it's a little rushed in the end. But today is all about coding actually, and since we already know how it works, and it's about the analysis, how do we make sense of the data coming out of the spatial transcriptomics technology. But before we move on to today's agenda, let me try to address some of the, so someone had a question in chat. Okay, yeah. So yeah, it's really hard for me to switch around with a small screen, but yeah, let's deal with it. So let me address some lingering questions from yesterday. So Arash is a postdoc from Payman Golshani's lab here at UCLA. Actually, it's a really cool lab. You're interested in neuroscience. He raised a question about HCR fish. So we talked about merfish, we talked about SlideSeq, we had another paper about star map, but he was asking what's the difference between HCR fish versus merfish? And I didn't know the answer, obviously, because I haven't read the paper. I thought I didn't read the paper, but actually he sent me the paper and I saw it. I actually read about this before. It's a really cool paper if you're interested, and I put the title here. So about the HCR fish, what's relevant to this class and is in its figure 1C. So I actually made a screenshot here only for that panel, figure 1C, where they show their barcoding strategies for imaging or do fish on gene expression. So so each row is a gene name. So let's count how many genes are there. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Okay, so around 16 genes they imaged, and they split them into, it looks like 6 different imaging rounds, and they show what are the barcoding scheme. I think dark means as a black or I guess either black or gray means it's light, and that gene should be light up in that imaging round, and blank means nothing or darkness. So yeah, how does this compare with merfish? That's the question. So I guess you can immediately appreciate, and there are fewer genes here. Merfish can do more than 100 and up to actually more than 1,000 in some cases. Here are just 16 genes. That's one difference. The other is merfish uses Hamming distance. So binary code, but both uses binary code, but here it's a 6-bit code. Merfish is longer, and you can also focus on the difference between any two genes here. Some genes share the same bit, and but they are most likely every 2 genes you look here are different by 2 bits. So you could call it Hamming distance 2 barcoding system or something like that, versus merfish is like Hamming distance 4. Another interesting thing is, so if you focus on just 2 genes, this SLC17A7, Can you see my cursor here? Yeah. So this gene is exclusively light up only in the last bit, in the last imaging round. And so did the SCAT2. So these two genes is only imaged in the last round, and this last round only imaged these two genes. So why is that? Because for one thing, these two genes are really important. They represent really important markers in the neuron, different populations of neuron. The other is they are very likely to be highly expressed, and you don't want them to contribute to other channels. So they represent a significant proportion of other channels, and they tend to mess up things. It's just a special consideration when you actually need to design things that you need to take care of. So what, and one last bit, I guess, what are these two readouts? So readout B2, readout B1, 488, 647, I would guess that they represent two different colors. So the numbers just mean its wavelength. It's about right, 488 nanometer or 647 nanometer. So two different colors in each imaging round. So you can actually resolve more than 2-bit. That's what this is. So yeah, I think that's a nice recap of what we did. Okay, so someone asked, what about the gray boxes? Yeah, gray boxes represent different colors. So in each imaging round, there are actually two colors. So instead of one imaging round for 1 bit, 0, 1, it's actually one imaging round for 2 bits, where each color represents 1 bit, if that makes sense. Let me know if you have, if you are still confused whenever.
Finally, one quick question, just to be clear. So yeah, this technique, the cells that are expressing SLC and GAT2, they cannot be distinguished because they're both, or they can be distinguished because they have two different colors associated with them, right?
Yeah, they can be distinguished. Yeah, two different colors, exactly.
Whereas if it was only single color, then we wouldn't be able to.
Right. For single color, you need two different imaging rounds. So the number of colors and the number of imaging rounds is kind of interchangeable here. So they could have switched the gray boxes to create their own columns, if that makes sense. To the right. So instead of, so let me make a rough plot here. If you can see the blackboard. So here they are kind of two matrices stacked on top of each other. Those top one is just black. The bottom one is gray. So what you can do, it's the same thing, that if you shift the gray box or everything related to the gray color down here and call all of these 0, all of these 0, it's the same thing, if that makes sense. But okay.
Thanks.
Yeah. Okay, so yeah, let's move forward. Quick recap. How do we go from raw imaging to the cell table? So we take multiple rounds of imagings and we count spots, and we summarize the information in a spot table where we know their locations and whether they light up in each round of imaging. So that represents bit 1 up until bit 16. Then using those barcodes, we can resolve their transcript, which gene they are. So it turns into a transcript table, and then we summarize it locally and assign those transcripts to individual cells, so we know for each cell where they are and how many genes for how much they express. Okay, and today is all about how to make sense of the data, and we did that a little bit. So let me just give you an overview, and let's dive into the details. So here's the data we are starting from. So it's a data table with 2 pieces. For each row, there's a cell with their location, X and Y coordinates, and their gene expression level. It's an integer matrices with a lot of zeros, and the integer represent for each gene and each cell, how many spot we detected for that gene. So we kind of went over this a little bit yesterday. So if we focus on X and Y coordinate and look one gene at a time, you can plot their spatial distribution. So what else can we do? So one thing to do with is to use the power that for each cell, we can measure multiple different genes. In the data set we had, it's actually more than 400 genes. How about we use the information of all 400 genes or so? And how do we make sense of it? We could do dimensionality reduction because that's a lot of genes, and we reduce it to a smaller number of components so we can make better sense of. So we'll talk about it today a little bit, and you'll have some practice. Yeah.
Sorry, just so are we just gonna assume that a lot of these genes are correlated with each other? Expression?
Great question. Yes, they will be correlated with each other. Yeah, if you think about it, why genes are correlated with each other, because it's often not one gene that performs function. It requires a couple of genes to make proteins, and they act together. Yeah, a lot of them will be correlated. That's one technique people often using even before spatial transcriptomics. And what else can we do? We know cells form types. Neurons are different from muscles, and neurons have subtypes. And how do we identify their subtypes? According to their gene expression patterns. Then we can use the genes and gene features to cluster those cells. It's called clustering or cell typing. And so we'll talk about that a little bit today. And once you get the clusters, what we can do is to compute this cluster centroid for each cluster across all genes. Then you kind of get a pseudo bulk RNA-seq profile. What we mean by pseudo bulk is it's like you computationally pulling the signals from all cells that belong to the same cell types and do some experiments, bulk RNA-seq on it. It's equivalent to that, more or less. And then finally, we want to use spatial information and how they entangle with their molecular information. So let's do a little bit about that. So if you have the cell type and you know where they're located, one question is we could examine the microenvironment to know for each cell type, are they tend to be neighbors of the same cell types or a different cell type, or what's the enrichment patterns of cell type interactions? Are some subtypes of neuron always be near blood vessels or near oligodendrocytes, and these kind of questions? Okay, so that's all about today's work. So four main points and all about how to derive analysis results from this measured data. So I kind of want to organize it into 4 coding sessions. So for each coding session, I'll do a little bit of talk, hopefully minimal, and then some type talk, or I'll be typing and do a demo on the simulated data. So you get a sense of how to apply this thing to actually, how you actually work, how do you actually go through things to review the thought process when we code. And then the exercise, it's on you. So you apply the same thing or similar thing, but on a different problem, on the real data we had yesterday. So here's the link to today's Jupyter notebook. I already sent it to you through email. And then let's post everyone's results to a shared document so we can learn from each other and have some fun. Yeah.
Is this different from yesterday's?
Yes, it's different. But yeah, I want you to keep yesterday's as well. Let's start from yesterday's actually. So and repeat. So and finally, your questions take higher priority than any of these. And please be interruptive again, like you did yesterday. Okay. Okay. So First of all, the first one is just a review. So actually, if everyone still have their yesterday's workbook, let's pull it up right now. I kind of did a demo halfway through and it's kind of rushed. So the first one is to try to reach this point by plotting this one gene, its spatial distribution. And we talked about a few points yesterday, and let's see whether you can replicate it and whether you can move further along, okay? So a few points are: be mindful of data point crowding. If the data set data point size is too large, it crowded everything, and you want to put a cap on the expression level. And we haven't talked about normalization, but let's do it today, okay? I guess first off, let me give you 5 minutes to start where we left out yesterday, if you still have yesterday's workbook. And at the end of 5 minutes, I want to have everyone to put whatever result you have to this shared Google Doc, okay? And then we move from there. That's gonna be the first coding section for today. Any questions? If you lost your yesterday's workshop, a link to the workbook, just let me know and I can send it to you again. I guess it's also in your email from yesterday. And also let me set up mine so that we could do something about it. Finish it. Okay, while you are doing this, I'm also doing the same thing, hoping to catch up where we left off yesterday. And one thing is you have to re-upload your data sets, which is inconvenient, but it was the same data you downloaded yesterday. So I'm running through this problem. Okay, so that's where we left off yesterday, and I kept all the codes. I just rerun it now. So that took me there. So at the end of the 5 minutes, I'm just gonna show you how you can do this. So you could just drag this plot to your desktop, and then drag it back to the Google Sheet. There you go. Then there's a plot. Okay, let's wait for 1 more minute. Then whoever has some results, let's post it here. Okay, yeah, I actually see a lot of. Okay, great. Okay, yeah, it looks like they're having a lot of. Okay, so just to recap, so we were dealing with this issue of outliers, where very few gene cells express high level of this gene, which skews the distribution to the extreme. Then one way we dealt with it is to put the cap on the maximum color, maximum value you can put color on. That was one way. So today, let's talk about actually another way, which is normalization. And it's also very commonly used in single-cell RNA-seq and also in single-cell transcriptomics. So what's the idea here? So the idea is because you have this orders of magnitude difference in gene expression level, one thing we actually commonly do is to take log of your data. By taking log, I mean, so if you take log 10, 10 will become 1, and 100 will be 2. So let's try this. So if you do, let me actually check myself. You could take log 10 on 10, it gives you 1, okay? So on 100, it becomes 2. So it makes the large number much less extreme than linearly does. So every fold change will be linearly represented. So let's apply that to our data. So we could try to take log 10 of our raw gene expression counts. So and try to do that. Once you if you do that, you see some error, negative infinity. That's because you can't take log on zeros, because log 0 is negative infinity. So what everybody often does is you add a pseudo count 1 plus 1 to all the expression level, and that makes everything works. So let's call it a new gene expression or normalized gene expression, NGEXP for now. And we can re-examine its distribution. So yesterday we made a box plot on the raw expression level, and let's do the same thing on the normalized one. Now you can see it makes much more sense in a way that it's well behaved, and we can make a histogram on it. Okay, let me actually separate these two things. The hist plot NGEXP. I did it twice. Okay, so histogram is more obvious. Now you can see what's going on in the expression level of this data. A lot of zeros and very few, but compared to the number of 0, but still many that are above 0. And you have some ones, some 2s. You have larger populations of 2s than 1s. It's kind of bimodal distribution, if you will. But what does it mean now? So now 2 actually means 100-ish, because you already take the log 10. But 500 is kind of less than 3 still. So it squashes the whole distribution to look much more well behaved. For example, if you still remember yesterday's result, this is the histogram now. Let's compare it with the histogram from yesterday, which is without the normalization. What does it look like? It looks like this. It goes from 0 to 500-ish, and you barely see all the high numbers because there are very few of them and it's extremely large. But after taking this log transformation, log+1 transformation, you kind of squashes everything down to something that's more reasonable. That's one trick, and people like to use it in gene expression data, also has some biological argument in it. That's because when you take log, you measure things in fold change instead of differences. So what do I mean? So if there are two cells, one cell expresses 2 copies of that gene, the other expresses 4 copies, it's an increase by 2-fold, right? But the difference is also 2. If you have another situation where one gene expresses 100 copies, the other the other cell expresses 200 copies, it's still increased by 2-fold, but the difference is actually 100 instead of a small number. So are these two equivalent if you measure the difference? 2 cases. Actually, let me write it down. So you have this situation where you have cell 1, cell 2, and gene 1, say gene 2. Doesn't matter. And so for gene 1, the expression pattern is 2 versus 4. Yeah, let's make this. And for gene 2, you have 100 and 200. Okay, so the fold change are the same, but the differences varies by a lot. So do you want to measure the differences between cells by fold change or by differences? In many situations, we would argue that fold change is more important because you could think about all cells share some baseline level of gene expression, and different cell type tweaks deviate from that baseline by a little bit. But what matters is the concentration differences and whatever relative to the baseline, and how much in concentration of that gene expression relative to the baseline. And that's measured arguably better in this way. So because of those reasons, it's legit to take a log normalization, just keep in mind. But yeah, let's apply it. If we just take log normalized counts and replot it, so just copy this thing. Okay. Previously, the color is by the unnormalized gene expression. Now let's color it by the normalized gene expression. And we remove this cap on the maximum value a color can represent. So it represents all the values in this. It naturally just shows this way without any cap on maximum color, something like this. Okay, so so that's a second way of making this plot look better, if you will. The first one is to remove outlier. The second one is to normalization that actually makes the outlier less obvious. Okay, so I'll stick to this. And one final pieces from yesterday is how do we do a rotation? Because I already wrote a function of rotation to you, it's just a matter of how to use it. So let's go back to the top. So here, there's a little function I wrote called rot2d, rotation for 2D coordinates, where the inputs are x, y, so x and y coordinates, and theta. Theta is the degree, the angle by which you want to rotate your x and y's. And then the output are also two things. So it should be the x after rotation, y after rotation. Okay, so let's try to rotate this to make it level. That's going to be the last task of the first demo. So what do we do? So we have our x and y. We know their coordinates that looks like this. We just send it to our function, okay? Rot2d x and y and theta. So the question is, then the results will be the rotated x and rotated y. So x r, y r. So yeah, then the question is, what's theta, right? By how much degree do we want to rotate this to make it level? Okay, any idea? The way I would like to solve about this first is by brute force. Just try it. Try to rotate it by some amount, okay? And see how it works. So let's say we want to rotate this whole thing by, say, 45 degrees, because that seems to be a lot. We will notice the difference. So then we replot this thing, but instead of plotting X and Y, we replot the rotated version of X and Y. XR and YR, you see? So we first rotate X and Y and we get XR and YR, then we plot it and keep everything else the same. Okay, so if we do that, let's see what happens. Actually, it rotates back pretty well. So yeah, so let's try another, try a different angle, say 60 degree, what happens? You see it overcorrects itself. It's rotating from here and in the counterclockwise fashion. And if I put 60 degree, it moves here. So it's actually working. Okay, so how do we decide which theta to do? So that you need a for loop. This serves as a coding review, I guess. Let's make a for loop and try different thetas and pick one that makes most sense, okay? So I'll call it for theta in a list of values we want to try. Let's go from 10, 20, 30, 40, 50, 60. 60 we already know overcorrects. Yeah.
Can you explain what NGEXP is?
Oh yeah, it's the normalized gene expression because it's defined here. Yeah, log normalized genes. Yeah. Yeah, very, yeah. Okay, so we made a for loop. We want to try for different theta value and how we rotate everything. Okay, you can also try it on your own workbook. But what happens is, so actually, let's count. Let's put a title on top of everything. So let's for each case know by how much we rotate it. Okay, by 10, we don't feel it much. By 20, by 30, by 40, more and more, 50, overcorrect 60, okay? So you can see its effect. So for now, let's just pick 40 and stick to it. Another thing you notice, that's going to be the really final thing, I guess. So once you, so you can see that the brain gets squashed a little bit. That's because the X and Y axis has different intervals by default, right? So if you count, maybe it's most obvious here. If you count from 0 to 2000, it moves this much on x-axis. And so 2000, and 2000 is actually this much on the y-axis. So that's why the X and Y are different. So the brain got squashed a little bit. So to resolve that, we'll just need another command to force it, to force X and Y to have the same aspect ratio. So that's called set aspect equal. And rerun everything. Okay, so then from 10, let's see the result, 30, 40, 50. Now it looks like a brain, like an actual mouse brain, which is wider than taller, okay? Yeah, that's gonna be everything for making a scatter plot, a gene expression plot in situ. Okay, let's pause here. Any questions? that's actually the warm-up for today. So let's now switch to the topic of today. Actually, so all of what we did is summarized by one function that I handed over to you today. So you can just run it and it'll generate this, okay? And you can obviously apply it to different genes. So SLC17A7 is just one example. Then you can apply it to other genes. Some genes will have very different spatial patterns. SLC17A6, it's only off by one number. It actually represents a different neuronal population, and GAD1 is for inhibitory neurons, the different population. And these markers down below are for glias and other stuff. Okay, so that's the first thing. So let's switch to the second. So we'll do 4. That's the second we're going into, and maybe after the second, let's take a break and do the other second. So the next topic is about dimensionality reduction. So we'll talk about 2 techniques. One is PCA, one is UMAP. PCA, or principal component analysis, it's simple, linear, clear, robust, and fast. As everything we know everything about it very clearly and for a long time. And it preserves the global structure or everything as much as possible. But it's a linear method. And UMAP is on the, in contrary, is very complex. It's nonlinear, very flexible, and slower, but reasonably fast. And it preserves more local structures instead of global ones. So we'll go into that by some examples. First of all, what's dimensionality reduction? Okay. Dimensionality reduction claims that if you have a big matrix, you can compress it to a much smaller one without losing much information. So in our case, you have n samples or n cells, a lot of genes, p features, p is number of genes. You can compress it to a much lower components. So in single-cell RNA-seq, you have n is about 1 million cell. P is about 10,000 gene, K is about 10 to 100 component. In spatial transcriptomics, in our data set, P is about 500 genes, and K, let's say, is about 10 to 20, which we'll do in a few. But how is this possible? I think someone already asked. That's because, so we had some assumption behind this. If this matrix is truly random, you won't be able to compress it at all without losing significant number of amount of information, because there's no free lunch. But luckily, biology has structure that means genes co-express to form functional group. There's a lot of correlation in gene expression patterns. And from the information perspective, it means redundant information. So genes that are regulated up and down together, in biology, it's called forming coordinate organization or whatever processes. But from the information perspective, it's redundant and we can remove those. And in other words, data lives in a low dimensions embedded in high dimensional space. High dimension is set by the number of genes you have, actually, but cell types do not randomly express all possible combinations of genes. They actually express a very subset, very modular modularized amount of genes, and in some accurate proportions. So that allows us to do the tricks we will apply now. So how does it work? Actually, how does PCA work? Here's a toy example, right? So let's assume you only have 2 genes. You have about 100 cells here, just 2 genes. Now you think 2 genes are too many. We want to reduce it by 1. We just need 1 gene, not 2 genes, or 1 component. It could be a combination of 2 genes. And the question is, or the same data can be represented by the data table where 1 row is a cell, and for each cell you have 2 gene expression, 2 columns. it's asking if you want to drop one column, which column do you drop, right? Intuitively, we know the answer. We want to drop the second column. That's why that has less variance, less information along it. Even if without it, we still have a lot of structure kept in this way, okay? So that's the strategy. So to codify it, the strategy is to remove the low variance column, okay? That's the naive thing we come up with. But how about now? Now the point is shifted a little bit. So if I ask you, which column do you want to drop? The answer is likely still the second column, but you probably feel some uneasiness of dropping it because dropping it just looks like this. So this is the original data with 2 genes measured. Now you just get rid of the second one. Everything claps onto this line. Okay. So what's a better strategy? So I think everyone has the intuition here. If we want to remove dimension, one dimension, we could do better, something better like this, right? So let's rotate everything by a little bit and define this line as the one component you have, and project every point onto this one line. And this line seems to preserve as much information as possible given our constraint. So a better strategy is to remove the low variance column, but only after a rotation. Let's first do a rotation and then drop the column that has the least variance. That's an algorithm of how we can do this. So that's the idea essentially behind principal component analysis. So what it does is it reduces data dimensions by removing low variance spaces, but after a rotation. And it needs to figure out the best rotation that you can do in your original feature space, in gene expression space, to project data points. And the nice thing about it is all of the intuition is recaptured by a math theory or a linear algebra theorem that's well known and well characterized, called linear value decomposition, which says if you have a matrix, you can decompose it into 3 other matrices, but that 3 other matrices are much simpler than the first one, and you can also express this into the sum of components. So this is the first component, PC1, PC2, up until PCR. The R's component. And in each component, there's a basis. Basis means after your rotation, where are you going to define your axis and the scores. So the projection of every point to that axis. And a single number that tells you how important that axis is. And from the most important to the least, as one captures the standard deviation or the variance, if you will, along that axis. And then you can drop the component with the least importance or least variance along it. Okay, that's spatial, and it works in more than 2D. I just show you 2D because it's easy to visualize in 2D. In reality, gene expression, if you have 400 genes, you can't visualize all of them, but you can still apply PCA and do a rotation in high dimensional space. So how do we apply it? Questions? Here's an example, gene expression data from the mouse brain. So you can try to evaluate any 2 genes and plot these. So each point is 1 cell. Then you see some structure, but every 2 combinations of genes you picked. So these are 2 different genes, these are 2 different genes, these are 2 different genes. You see different patterns. But the question is, what's the overall patterns among those cells that we measured, right? So the idea is to plot PC1, the most important PC, the PC with the most variance among your data across all genes, versus PC2, the second most important one, and ignore all the other. Of course, you ignored some, but that's going to be the best 2 that we can pick up linearly. So if you do this, you actually find some structure that's very nicely from this data set. That's yes, a single RNA-seq or gene expression profile from the mouse visual cortex. And you can see all the points tend to align along a line. So it suggests some continuous changes among those cells that goes along a line. And it happens to, if you recognize the labels, it happens to align with what we know about those cells in anatomical fashions. Cortex are organized by layers, and the anatomical locations of those cells happens to be agreeing with their gene expression, the overall gene expression patterns. Yeah.
So when you apply PCA, you get two matrices, right? You
yeah.
Corresponds to the rows and corresponds to the columns.
Yes.
Is this plot the one that corresponds to the rows?
Good question. So each point is a cell. So I guess I'm plotting the cell by component, right? So yeah, what he's asking is, so so originally you have a cell by gene matrix. So you're actually decomposing it into cell by component and component by gene. Yeah, I'm plotting the cell by component matrix after that. Okay, so that's one thing. What else can we do? So that's the demo I'm gonna do for now, for today. So you can just apply it to rotate the mouse spring. So I just show you how we can apply rotation to the mouse spring so that they look level. But I did this by trial and error. I tried many different rotation angles and picked the best one. What if we don't want to do that? We just want to solve for the best angle to rotate things. Actually, you can just do PCA on this XY coordinates of our cells. So this is the original coordinates you get from some data set. Say they have very different orientation by default, but then you can apply PCA and plot PC1 versus PC2, right? Then more or less everyone agrees with each other. Because why is that? Because mouse spring are wider than taller. So the first PC are the principal components that captures the most variance in your data. The most variance is obviously this left to right thing. So by applying PCA, you immediately identify the left and right. Where are the left and right? And where are the top and bottom? Okay, so I'm gonna show you how to do this. And then with the same idea, with the same code, you can apply it not to just X or Y coordinates, reduces the two components to 2, but you can apply it to all of the genes and reduce it, say, to 20 PCs and see their structures. Okay, that's going to be on you, the exercise, and on me, it's going to be the demo. So that's for PCA, but let's move on to another technique called UMAP. UMAP is a much more complex thing than PCA. PCA is just a rotation, linear rotation. But UMAP try to, what it does, it first represents high dimensional data points by a neighborhood graph. So they first capture for each point, what are your neighbors? And then they want to project it down to 2 dimension while keeping the neighborhood structure as much as possible. So the second exercise on you, so I'm gonna apply UMAP as well. But the second exercise on you is to apply UMAP on this real data as well. So if you apply it on our gene expression space, you'll get something like this. So you see some structures, and if you color it by gene expression, you kind of see that the gene we picked is highlighted kind of in one of the groups or one or a few subset of groups than others. So it's picking up the similarities among cells in their gene expression patterns, okay? So that's 2 exercises that I went over. Next, let me do a demo on how to apply PCA. Yeah.
So is this the same data set as yesterday?
Yeah, same data set as yesterday. And now I want you guys to switch to today's Jupyter notebook. Again, make a copy, run it through, and work from there. Okay. I'm also going to set it up from my side. Questions from the from the Zoom? Okay, first. Okay, I need to run this. Okay, let's get started. I got mine set up. It's a collab day 2 crippled template. Okay, you still need to upload the same data set there in order to run. So the first thing I'm going to show you is how to do PCA actually using Python. You don't actually need to re-implement all the math because someone already figured it out for you. You just need to figure out how to use those tools correctly using some packages. So the package we are going to use, which I already included here in the first few lines, it's called, let me copy it here. Copy it over so you can see. It's called sklearn. Sklearn is a package for a lot of machine learning related tools. And specifically, we want sklearn.decomposition PCA. So PCA is the thing we want to use. So how do we apply for, how do we do this? So one thing is to go to their documentation. Let's say sklearn decomposition PCA. Immediately we can see how to use it. There's a lot of things on it, but we know that the key parameter is how many components you want to you want to use, or how many dimensions you want to reduce your data set into. So in our case, let's say we apply PCA and components equals 2 because I want to only show 2 components. Let's call it PCA. Okay. And then we go back. What we can do with it, because I know this already, I've applied for a long time, but just want to show you a little bit. Here's a command called fit, or here's a command called fit_transform. Either way works, but I'll apply this fit_transform. So we do PCA.fit_transform, and then I want to do it on our X and Y coordinates, just as a demo, but you want to do it on other things later. So let me copy this. So we have our X and Y coordinates already, but they are 2 one-dimensional things. They are 2 vectors, if you will. We want to apply PCA to a matrix. So we just reorganize. We basically want to stick them together into a single n×2 matrix. So let's reorganize it a little bit. I'll use a variable called xy, which is a v stack of X and Y and transpose. Okay, now I created a cell by 2 matrix, number of cell by 2 matrix. Let's show what XY looks like. So the first column is X, our original. Second column is Y. That's all what this does. So it's a little bit of math, but for now it's not that relevant. What I did is just put the first column, X into the first column, Y into the second column, so I can apply PCA to this joint space of X and Y. So and the results will be XY results will be PCs, so the principal components of X and Y. Okay. And if I do that, here's the result. So it gives me back 2 columns as well. So the input is 2 columns. It gives me back also 2 column. Why is that? Because I assigned 2 to be the number of PCs I want to use. Okay. So next, what I want to do is to replot the principal component. So I already give you this function, this little function, which was the progress we made yesterday and earlier today as t-scatter. It's a scatter plot. So you could plot X or Y like we did yesterday, but now let's plot the first and second components of our PCs. So everything, so we select the first row, first column of PCs and the second column of PCs. Actually, to make it more clear, so PC1 equals our PCs, the first column. PC2 equals PCs, the second column. PC1, PC2. Let's compare between these two. Okay. So the first one is, so this is the XY coordinates. The second one is PC1 and PC2. Okay, let's see what are the differences between them. Okay, so this is the first one. This is the second one. Okay, so so intuitively, what PCA does is it rotated, but also flipped the original the original frame sections, right? Why does it flip it? Because PCA only finds the axis with the most variance. It doesn't define a positive or negative direction, if you will, in your axis. So that direction is a kind of arbitrary. In other words, you can put PC1 negative PC2. Negative PC2 is still PC2. If you do that, that gives you back what a normal brain that we think it should look like. Okay, so basically what PCA does is it automatically finds a rotation that makes your data looks better, or where the first component has the most variance and second component has the second most variance, okay, without knowing the angle priorly. Okay, that's what PCA does. What else? What's one more point about PCA, which is we know for PCA, we know the relative importance about different axes. So now we have 2 axes. We want to know their relative importance. That can be captured by something called variance explained. So let me go back to this document and look for its attributes. Attributes is this thing that I don't know how to. So when you run it, it will generate these attributes, and we are specifically looking for something related to variance. So here is one term called explained variance ratio. Let's use that and talk about that in a second. First, we apply it to our code. We do PCA.explain variance ratio and see what it looks like. It's just 2 numbers. first number is 0.67, second number is 0.33. What does it mean? It means the first principal component captures 67% of your data, of the variance in your data. And the second captures the rest 33%. So now you know this left and right lateral thing captures 67%, and the top and bottom captures 37%. And this will be extremely useful when you use when you apply PCAs on a large high dimensional data, say apply it to 500 genes. You want to know the relative importance of your first PC, second PCs, and so on and so forth. Okay, so that's PCA. The next thing we talked about is UMAP. So UMAP is, application-wise, it's very similar. It's also a package people wrote about. You just need to learn how to use it. So in this, I already import from UMAP, import UMAP, capital UMAP. So I'll just paste it down here. Okay, and actually they designed their function in a similar way that in applying PCA, so you can actually just copy this, literally copy this down here. Okay, so instead of doing PCA, now we are doing UMAP, and we assign it to UMP, say. And you can do UMAP.fit transform on your, I'll still apply it on our X and Y data, although it's kind of silly, I know, but I'll do it. And the results, I'll call it UC's UMAP components. And let's check what the UC does. I know this is going to take a while, about 3 minutes. Let's wait for it to continue, because we have a lot of cells and UMAP requires a lot of calculations. But at the end of it, I'm going to do the same thing. I'm going to plot its UMAP component and see what it actually does. So again, use UMAP component 1 equals our resulting matrices with the first column, and UMAP component 2 is UC, the second component. So Python always counts from 0. So the first one is the zeros one. The second one is the first one. It's a bit confusing, but let's do that. Okay. So that's that. While we are waiting, let me just share with you one more thing about dimensionality reduction that kind of captures everything I want to talk about it. So let me reshare you with your, then we can switch back after it has been finished. Okay. Okay. So we talked about the PCA is simple, it preserves global structure. UMAP is very complex, preserving local structures. And what does it all mean? So one thing is to understand the math behind it, how you generate it. But another thing is to apply it to some toy models to gain some intuitions of what these kinds of transformation is doing, right? So that's what I did here. So I created some simulated data, which is points along the range. So you can think about it as cells that with two genes being expressed, but these two genes happens to be, their expression pattern happens to be so peculiar that all of the points is along a ring with a small opening. Okay, so in this 2 dimension, we can visualize everything. So what if we apply PCAs to it? That's the result here. It does nothing to it because PCA only apply a rotation. It actually does something. It applies the rotation, so it knows the existence of opening, has some differences in their variance along different axes. So this is the maximum variance. This is the second most variance. So, but basically, it's just a rotation of this ring, right? And what UMAP does, it completely destroys its shape because it preserves the local neighborhood. All it knows is cells tend to lie along a ring, and it tries to preserve this one-dimensional structures that that cells are lying upon, but it doesn't care the shapes or the wiggles in this. So this is kind of arbitrary in this sense. So, but it's unfair for UMAP because UMAP is designed to not preserving global structure. Another informative way of looking at it is to plot things versus angle. So we know that the data structure, so how many dimensions are there in this data? We know there's only one dimension, which is defined by where the angle of the cells. So there's only, so it goes from 1 to 0 to about 360 degrees. It's a circle, right? So everything is along an angle. That's the one dimension you want to extract from any linear dimensionality reduction algorithm. You should pick up that aspects and know that. And then you can plot the angle, PCA versus angle. Then you see something like this. So what's good and bad about it? Good about it, it's a nice shape. What's bad about it is for the same PC1, you have many different, two different angles. So if you just focus on intersecting this with one straight line, one vertical line like this, it means for the same PCs, you have two different angles collapsing onto each other. It means PCA reduces information by a lot because it confuses two different angles as the same thing. That's the information you lost from PCs. Whereas in UMAP, if you apply UMAP on one dimension and you plot it with angle, this plot doesn't seem to make sense, but it looks like you have a one-to-one relationship between UMAP and the angle. It means every one angle is represented by a unique UMAP component. It preserves the angle in some ways and knows its local structure, although it's rigged a little bit. Okay, so that kind of summarizes what PCA and UMAP, the strengths of each thing and the disadvantages of each thing. So I think we should have finished this. So this is the result, if you still remember. But this is the original feature space. This is after applying PCA. It's just a rotation, preserves everything up to a rotation. This is after applying UMAP onto it. It's a warped brain, if you will, because it still preserves a lot of structure. It expands somewhere, it concentrates somewhere. It's doing its complex things inside. Okay, that's PCAs and UMAPs. So the task for you is I've applied these kind of things only to XY coordinates, which is silly. What we actually want to apply is to the gene expression space, to the features you want to. So you want to do the same thing, but on a different matrix, on the feature matrix, which I already get it for you. So it's in our data, apart from the first two columns, all the rest of the columns are some gene expression. You want to send this as input to PCA to UMAP and see what it pops up. Okay, so I'll give you guys 5 minutes for this, and at the end of the 5 minutes, please post your results up there and see if we need more time or anything. Okay. Uh-huh.
1 and 2.
Oh, 1 and 2. Yeah, both PC and UMAP. They kind of seem similar. While you're doing it, I'm also doing it if you want. So by the way, I kind of know this class is very diverse in their coding skills and everything. For some of you, you may think this is too simple, and for some, you might not be knowing what we are actually. You may not catch up with the speed. But let me know if you're stuck at some point, or you can just try to watch. Me coding. So. Okay, I got my results. Actually, it looks like this after applying UMAP to the gene expression data. And now my results for PCA for this is like this. This is, by the way, PC1, which is the x-axis. PC2 is y-axis for all of the cells, colored by the expression level again of SLC1787. You see that SLC1787 is actually expressed in the PC1, actually highly correlated with PC1, but not PC2. And this is PC3 versus PC4 and UMAP. Okay. Let's check back here. Okay, I see something like this, which looks interesting. Okay, someone gets this point, PC3 versus PC4, which kind of like the one I got. So I guess hopefully we are both correct. Oh, and someone gets this, which is also kind of like the plot I got here. Yes.
Is the you play UMAP, it could always be like rotated around.
Yeah, always.
So
Yeah.
like all of these are
Yeah, exactly. Yeah. Okay. So 2, I see 2. Okay, I think you guys are starting to get to this get this idea. There will be some details here. It takes some practice to actually code efficiently and know what you're doing, but that's the essence. Let me just review it a little bit. So for PCA, you actually just run one command if you know how. Then the only parameter, so you need two parameters. One is to tell it how many components you want to extract from. So in this case, I put 20. And the other one is the original input matrix, the one you want to compress. So that's the feature matrix. You could do feature stop values of features, it doesn't matter. Then the results is send it to PCs, this a variable called PCs. And if you check the dimension of PCs, you actually see it's the number of cells by 20. So you reduce it from about 500. So actually, let's compare between these two. So let's compare between the dimensions of PCs, which is your results, versus your input, which is a feature matrix. So feature matrix has 483 columns, but the results has 20 columns. So it does its job. And then you can plot it. Okay. And you also see that UMAP actually shows its power. In this case, it tends to show at least a lot more features than PC1 and 2. That's why people use it. Although it's harder to interpret what's going on inside. That's a different problem by itself. Okay. Any questions? Okay. We are about 3. So let's go into our last two topics. My goal is to finish today by 3:30-ish. That's the hope, but I know everyone wants to have an earlier break in Thanksgiving. So normalization, we touched upon it. Why do we need to do normalization? So for one thing, we notice some, it's less obvious, but there's some strange things in UMAP, which say on this branch, there's this continuous pattern along, which happens to coincide with, with the expression level of the single gene SLC1787. So it's highly expressed here, it's lower expressed here, and you have some continuous pattern. So that often suggests, but not always, some known technical artifacts of transcriptome measurements, either single-cell RNA-seq or FISH data. The reason is that you have cell size factors in all of those measurements. For example, cells could be bigger or smaller, but their overall proportion of gene expression level could still remain the same. So cell could appears to be bigger or smaller. That's by bigger, I mean having more RNAs captured, and smaller means less RNAs captured. But that's not, that's a technical artifact rather than that a biological one, most likely, because in sequencing, you have a step called amplification, which for different cells, you could end up having different ratio of amplification. And for spatial transcriptomics, you cut your brain section, say, tissues by a thin slice. In that thin slide, maybe only part of that cells is captured. Then you lose another half. So the overall proportion of cells, of transcript, may not be that meaningful. So by normalization, we can remove these kind of artifacts before we do any downstream analysis. So here, so here, what do we do? So we apply this normalization, which I will show you now. But after applying it and redo UMAP or whatever analysis afterwards, you actually see quite big differences. So this is before the plot we got just now, and after normalization. You actually see even more features. It's hard to tell from directly UMAP what it's actually doing, but at least it's a lot more feature than previously. Okay, so that's exercise 3. It's a small one. I'll probably just do it and show around. Okay, normalization. Okay, what do I mean by normalization? So we started from this raw data called feature matrix, which if we look into it, it's an integer matrix, always an integer, because it's a number of counter spots for each gene in each cell, with a lot of zeros. One thing we want to check is we take the sum along each row to see the total number of transcripts we captured for each cell. Okay, so sum along the columns for each row. Okay, if we do that, we kind of get the total number of transcripts for each cell, one for each cell. Let's see what it looks like. So let's make a histogram of it. They look like this. So some cells have no transcripts captured, some have 500, some will have 1,000. So you see a large differences between the total number of transcripts that are captured. Why is this a concern? It could be because the tissue section, for example, you cut through half of the cell, cut through 1/4 of the cells, or for some reason the probes you're setting is a bit uneven, and then that affects some regions of the capture rate of our mRNA molecules in some regions more than others. So let's try to stretch everything to the same total number across all cells. So how do we do that? We normalize it. So we first take the sum, we call it the coverage of each cell. And we take the median of the coverage, which we call MEDCov. So yeah, that's the coverage I just showed you. What's the median coverage? It's 246. Does it make sense? It kind of makes sense. So 246 is kind of the median. So we want to normalize the raw matrix by this median one. So we actually divide the raw matrix by this coverage over median coverage. So if a cell have more transcripts than median, it tends to shrink the total, shrink its features by that amount. If a cell has less than the median, it tends to expand its features. So that's what we call the features normed, and then we can reapply this log 10 trick. Okay, so that's what it does, and it runs very fast. And then we can reapply PCAs and UMAP and stuff. It'll take another 3 minutes. That lets you to the point of this. Okay, like this. Okay. So that's normalization. The final topic of today, exercise 4, is clustering. Clustering, as shown here. Let me actually expand this. What is clustering? So it's an algorithm that groups cells by their similarities in gene expression levels. And in this, in particular, we are going to talk about just one most basic, very simple clustering algorithm called k-means, where k represents the number of clusters you want to identify from your data set. And how does, so what k-means does is actually very simple as well. It's an algorithm that repeatedly does these two things. In the first step, so initially, it randomly assign every cell a cluster label just by random. And then it iteratively compute the centroid of each cluster. And then they update the labels such that each cell was assigned to the cluster with the closest centroid. And you update and update. So each time you update the cell labels, you recalculate cell centroid, and then you reassign cells. It's like a chicken and egg problem. So how do you know which cluster? So how do you know? Say it this way. So if you know there are 5 clusters already and know their centroid position, you kind of know how to assign cells to each cluster. Just assign it to the closest cluster. But then that information helps you update the centroid locations better. So by iteratively doing that, it converges and generates some meaningful results. And here is the result after me applying it to the gene expression features, normalized ones, using k-means clustering with k equals 20. It goes from 0 to 19. So this plot just shows what are those 20 clusters. So each cell is assigned a color, and you can see that you can see some anatomical structures inside the mouse brain on the left plot. And you can see that in the UMAP space, actually, each cluster represents a small population. So the groups of cells with similar gene expression were assigned to the same clusters. Okay, so that's k-means. That's your task to apply it to the real data. But before that, let me just show you how to do it on a demo data, on a much simpler one. Oops. Did I stop share? Yes. Okay. So k-means, how do we do that? So let me create a lot of blank spot. I'm going to generate some data set, again, with 2 genes with random numbers, okay? So I'm going to generate random numbers with 100 cells and 2 genes. Okay, here I generated 100 cells and 2 genes. That's my feature. I'll call it fake feature, maybe FF. I'll just call it FF anyway. So, but I want this to have 2 clusters, 2 natural clusters, if you will. So in the first 100 level, 100 cells belong to 1 cluster. The second one belongs to a different cluster. Okay, that's my FF. Okay, yes. And then the second one is shifted away from the first one by 10. So now it might not make sense to you. I'll explain in a minute and make a plot. So what's the shape of FF, for example? It's 100 by 4. Why is that? I'm confused a little bit. Okay. Oh, 104. So I stacked them the wrong way. It's a vertical stack. So you have 2 ways of stacking two kinds of matrix, either horizontally or vertically. So let's do it this way, vertically. So it's 200 by 2. What do I mean here? So I randomly generated 100 cells, and then again 100 cells, a different population. So 2 populations in total, there are 200 cells with 2 genes. So let's plot their distribution. What do they look like here? So let's apply this trick again. FF, the first component, FF, the second component. Let's make the point size a little bigger. Okay, indeed we see 2 populations because I intentionally created 2 clusters, if you will, 2 cell types. So next, I will show you by applying k-means clustering. Hopefully, if it all goes correctly, we recover these 2 populations. So all of the cells in this cloud should belong to 1 cluster. All of the clouds there here should belong to a different cluster. Okay, so let's learn how to use k-means. That's somewhere down below here. From sklearn, again, cluster, import k-means. Okay, we'll do that and use k-means and see what parameters it actually have. So let's search sklearn k-means. Okay, again, so the most important parameter is number of clusters, or the k in k-means. In our case, let's apply 2 because there are 2 obvious clusters. So n clusters equals 2. And again, if you scroll it down below, you want to use this model to fit your data. We'll in particular use this fit predict. Let's apply fit predict on this fake data, FF. And the results, let's call it clusters across. Okay, let's check the dimension of clusters. First of all, it's 200. Why is it 200? Because for each cell, there was a label. So it looks correct. And let's see what it actually has. Oh, plus. So it has a bunch of zeros, a bunch of ones. 0 means one cluster, 1 means another, a different cluster. Okay. Then next, how about we color this by their cluster label? Say we add a C, actually G expression. Actually, let's do something simpler. So yesterday we did plt.scatter, so we can apply this with C equals our clusters. Okay. Okay, so now you see two clouds actually have 2 different colors, each representing the cluster label of their cluster, and they seem to be doing its job correctly. I also, where is it? I also handed you a different function called plot cluster. Let's apply it. I didn't say what it does, but let's use it a little bit. Let me show you. So apply this plot cluster. It needs your clusters that we already get. X and Y, we already have UX and UY, which is supposedly the UMAP coordinates if you have X and Y, but I didn't apply UMAP on my fake data. So let's skip it or just call it X and Y. So just repeat the same thing twice and make the size larger. So, oh, I didn't run it yet. Oh, by X and Y, it's actually FF, the first column, FF, the second column, okay? Ooh. Oh, and again, X and Y should be the first column and the second column. Okay, so it's basically ignoring the second part because the second part is the same as the first part. But what's shown is again the same thing: the XY coordinates and the cluster label of each cell. Okay, so why do we want to do this? Because now, in order to understand k-means better, we want to tweak its parameters a little bit. I used I used 2 components. I wanted to generate 2 clusters because I know 2 clusters is the right answer here. But what if we force it to generate a different number of clusters? Okay, so let's tell it to generate 6 clusters instead of 2. What would happen here? Okay, so still you get a vector of 200. Each cell was assigned 1 number, but now you can see the numbers could go from 0 to 5, right? 6 different things. And if we replot everything here, what do we find? It's actually splitting each individual obvious cloud by some other criteria. We know in this case, then we over-clustered because we generated from random number, all of these clouds are equivalent from each other. But still, it will force it to to identify some patterns from k-means clustering. So that's actually a general caveat of applying clustering algorithm. You need to know a priori what's the diversity inside your data set, and you kind of need to test through a lot of things to see what makes sense, the best sense to your data. Unfortunately, in reality, the things almost always doesn't go clear cut like this, in which you immediately can eyeball it and see there are only two clouds. But yeah, you'll deal with it. So now your turn is to, so instead of using my simulated data, you're actually going to use the feature matrix, if you still remember this thing. And this whole big matrix. For each cell, you have these many genes, but you use this information to cluster it and see how many clusters, or you assign how many clusters to generate and see if the pattern makes sense. Okay, I'll give you guys 5 minutes on it, and if someone has some meaningful results, please post it online. And at the end of 5 minutes, let's see. If we have extra time, we can go over into some bonus points of more advanced analysis. Otherwise, we could linger longer on this point. Oh, wow. Yeah, nice. Yeah, someone got to this point. So yeah, from here you can see so quite a lot of information. So it's the same cell represented twice, first in their physical space, in their spatial locations, and then by their similarity in gene expression using all 500 genes or so. And you identified clusters among those high dimensional gene space, which is represented by different colors, and you can map it back to the original spatial locations. And you can actually see that for some cells, some cell types, if you will, they are anatomically form some specialized structures. So you can see this light blue cell types, which is probably located somewhere here, that goes along the outer range of this mouse brain. And here you have this sharp boundaries, that's hippocampus of mouse brain, that are similar to each other than their surroundings, and so on and so forth. Okay, so mine, I also tried to create mine just went through. So I guess one thing I liked about coding is, however hard it is, the first time you learn it, you can apply it every time in all cases. So we talked about PCA, we talked about UMAP, we talked about k-means clustering. When we are learning it, it takes a lot of trials and error, and you need to generate lots of lines of code. Once you get it, it's very simple. So actually, I summarized all three things in 3 lines of code. So the first line does PCA by applying normalized feature. You just get PCs. The second line does UMAP. You just get UMAP. And third line does clustering. Okay, then you can represent. You can plot it out with another line, which you figured out how to make a plot previously. Okay. Yeah, it'll take some time for you to get efficient on this, but yeah, that's the overall idea. So with this, what else can we do? So we talked about, so this is very abstract. It shows you a lot of information about similarity differences about and their spatial locations and stuff. Ultimately, we want to get back to the genes. Genes are what we measured and what we care about and for biologists. So one way to do this is, now that we have, say here, 10 different clusters, what's their gene expression pattern across those 10 clusters? Right? How do we visualize it? So that's in our bonus point 1. Let me just show you how to do it. It's quite straightforward. So the idea is, so we want to take the centroid of everything. So actually, let's start from 2 pieces of information. One is our normalized features, or the raw features if you want, but I'm here sticking with the normalized feature. For every cell, the expression level of every gene. The other is our cell type. For every cell, we have their cell type label. That's in, let's call it plus. For every cell, we have their labels, 1, 2, 3, 4, 5, etc., etc. What we want to do are 2 things. One is to merge all cells belong to the same clusters together. And then we take the mean for every gene. So it's the mean expression level for every cell, mean expression level of every gene across all cells belong to that cluster, and apply it to all the clusters. Okay, it's a mouthful, but in command-wise, it's not that complicated. So what do we do? So let's create a temporary data table that makes a copy of the original feature matrix. Then we're going to mess up with it. So let's first take a copy, so we keep our original version. So here, for every cell, you have their gene expression, but we want to add a new column to it called cluster label or cluster, something like, which we know already. We know its result. Just assign it here. So it becomes a larger matrix where the last column is their cluster label for each cell. Okay, so then there's a powerful command called group by in data manipulation. So group by means literally group everything by something. So we want to group this data matrix by the last column, by the cluster label of each cell. After you group by them, you take some operation. The operation we wanted to take is mean. Take the mean across everything. Then the results will be the centroids. What does it mean? Let me show you. Okay, and it's actually really fast. Here, because I defined the 10 different clusters, so the results is each row is one cluster. Each column is one gene. So it's 10 cluster by 483 genes. And the number shows the median, the mean expression level of all cell for every gene. Okay, so that's our centroid matrix. Now, this is a much more manageable size than the original cell by gene matrix. It's 80,000 cell versus our gene. Now it's 10 clusters versus our gene. Now we can actually visualize everything with visualize this matrix by a heat map. So we often know about, so let's make a heat map with this as an input and see what it actually looks like. Okay, so you see some patterns. So each row is one cell type, each column is one gene, but because you have so many genes, it's so packed. So let's change the figure size a little bit using similar trick we did yesterday. So we want to show figure size, make it wider, let's say 12 by 5, I don't know. Okay, so that looks better, but something still doesn't works as well as we wanted. One thing is we notice large variance across columns. So it could be a good or bad thing. That just means different gene has different baseline level of expression. So another thing to do is if you want to understand how different cell types here are different from each other, you want to highlight their different relative differences for each gene instead of between gene baseline differences. So we can do some improvement on it. One thing we could do is called z-score for every gene, of course. So renormalizing the expression range for every gene so that the cell type with the least expression is about -1, and with the most expression is about +1, and the mean is around 0. That's what z-score is doing. So how do we do z-score? So now we have the centroid. Centroid is here. We first subtract it by its mean across for each gene. So actually, how do we calculate the mean for each gene? That centroid's mean axis equals 0 or equals 1. We could check equals 0. So for each gene, we get its mean expression. So we subtract by its mean. So every gene has the same mean, which is 0, and divided by the standard deviation for each gene. So divide by the standard deviation for each gene. Let's see if this works. Okay, it actually works. So the results, let me call it z-scored centroids, if you will. Okay, and replot this heat map. So this heat map is useful, but in some ways, but not in highlighting the differences across cell types. So let's try to replot it by replacing it with the z-scored centroid. Okay. Oh, not defined. So I might have some typo. Z-scored. Okay. Okay. So yeah, now it does show a lot of differences, but because it's so randomly distributed and still doesn't make much sense. For example, if I ask you, what's the differences between cluster 0 and cluster 1? You can see a lot of genes are different between them, but it's, I guess, less organized as we wanted. Okay, and that, but before we do that, let's change a color map. So C map, and okay, so actually change a color map does show things better, because in this case, 0 is about neutral, is about white. And if it's higher than 0, it's red, and lower than 0 is blue. Okay, so yeah, what else can we do with it?
Hi.
Yes, go ahead.
We can't see your collab notebook. We can only see your slides on Zoom.
Oh, sorry.
No worries.
Okay. So yeah, do you see it now?
Yes, I see it now. Thank you.
Okay. Yeah, then we have gone quite far. But anyway, we started from here, clusters, and then we generate the centroid of each cluster, and then we're trying to play with it to make sense of it. Okay, so one thing we noticed is, so genes don't have to be ordered this way. So we measured 500 genes or so. We assign a particular order to each gene, but their order is kind of meaningless. So how do we rearrange its order? So to make it, I guess, make this genes with similar expression across cell types to be closer to each other. So one way to do it is to do a cluster map. Some error because it's some details. Let me explain it. Okay, so that's what it does. So so this heat map is the same heat map as this one, except we rearranged rows and columns. And because rows and columns are meaningless, we can rearrange them anyway. So now you kind of see what's going on here. So each cluster actually has a unique set of genes that are highly expressed only in those clusters. Cluster 1 has these set of genes, cluster 2 has these set of genes, and so on and so forth. In our case. So this actually suggests, the cluster we are picking up is actually making some sense because it's supported by a lot of genes and they are mutually exclusive. Yeah, that's what I'm going to talk about for clustering. Again, in the end, there was bonus point 2, which is about enrichment analysis of spatial proximity. I guess, so where we are at, Mara, do you still have a question or has that been resolved already?
It was resolved. Sorry, I forgot to put my hand down. Thank you.
Yeah, okay. Yeah, thanks. So that's clustering. That's how we pull genes, the central profile of each cluster. Yeah, finally, I don't think I'll demo it because it's close to 3:30 right now. But if you're interested, stay and I can show you later. But let me talk about the general idea. And I'll hand you the full solution so you can play around with it later. So far we've been playing with gene expression. We've been playing with spatial coordinates. So one unique opportunity of this data set is that we can combine them together and actually make sense of the local environment of the tissues. So one question is, are some cell types close to some other cell types in physical space? or are they enriched in local proximity in any ways? So one example of this is a recent paper, Fang et al. 2022, Science, figure 4, if you want to check it out. But how do we get at this question? So how do we know, let me reiterate it. How do we know that certain cell types are closer together in a higher frequency than random? That would be the way to put it. So one way to do it is to shuffle your data, to create a random thing to compare with your actual data with. So this is your actual data. You know how to get to this point, make a cluster and know where each cluster are. So then once having that, you can just count for each cell type what their neighbors are. Each cell for each cell type, what their neighbors are, and you arrive at a matrix for each pair of cell types, how many connections locally do they have? So the question is, are those cell types enriched, or are those frequency more than random? So how do you create a random? You just reshuffle your cluster assignment of each cell randomly. So to visualize it, it's the same number of clusters, both 20 clusters, right? But the bottom one is we reassigned every cluster randomly so that the brain is a homogeneous brain. You kind of dissociate every cells and rearrange them. But now it looks like this, and you can repeat this a lot of times, 1,000 times, if you will, and compare the random versus the actual data and see where each cell type pair light up. So what do we mean by that? So each row and each column is one cell type, okay? So then this matrix highlight how many times every pair of cell types are locally are neighbors or in physical space. So that's your observed data after I counted for 20 cell types. And you can randomize it and regenerate this matrix. That's your expected value by random. And then you can compare between the observed and the expected, say, divide observed by the expected and see if it's above 1 or below 1. Above 1 means it's enriched. Below 1 means it's depleted compared with random, right? Then you get something like this. The enrichment of every cell type pair in their spatial proximity, right? So that's a hand-waving way of doing it. And in the Jupyter notebook, you can try to play with it. And I'll show you how to do it. And I'll send the solution to you later. And another way is to do that. You can also test statistically whether the enrichment or depletion is significant, or is it enriched enough to be more than random chance. That's what something called empirical p-values and stuff. Okay, so that's everything I had. Last point is, so I kind of show you how to do things from scratch, from the most basic component. But obviously, there are people who understand things, who build pipelines and tutorials specifically on certain cell types. So I call it canned analysis tool. You don't have to understand what's going on inside. They apply complex things to specific data set. So there are tutorials and documents to this. One good tools. Nowadays, there are many tools. One good tool is called SquidPy. If you want to learn how to use SquidPy, they have detailed tutorials on how to analyze, say, imaging mass cytometry data, how to analyze seqfish data. For us, they actually have how to analyze VisGen data. And to apply this, you just need to run through their examples. You won't gain insights into how everything works in details, but you will quickly get some output and see the results. But I guess the philosophy of this class is I want to show you what's behind the scene and what's the thought process, what's the caveat, and what all the parameters you can apply to cells sorry, the data that makes it look different. So yeah, that's it. With that, that's the end of the workshop, and happy Thanksgiving. This is a reminder that please do this. Yeah, and I'll stay for a while if you have questions. Stop recording.